Good morning, everyone. Thank you for joining me today for the second webinar in the ACell webinar series. My name is Josephine Lambong. I am the Manager of Science and Industry Affairs at the Alliance for Regenerative Medicine. Today's webinar is on the topic of manufacturing of cell-based therapies. So over the next hour, you'll hear about the principles of cell therapy manufacturing, the different steps in unit operations, as well as process and technological considerations from two of our lead contributors in ACell, who I will introduce in just a few minutes. For those of you who are not familiar with ACell, it is a collaborative case study on the implementation of quality by design principles in cell-based therapies. This is a project that ARM undertook several years ago after feedback from our members about some of the CMC issues that the field as a whole is experiencing. It occurred to us and our members at that time that these issues were not unique to just cell and gene therapies, and in fact, some other segments of the biologics industry had gone through similar growing pains before. The example of the AMAB document was brought up, and it was something that was instrumental and one of the things that allowed advances to occur in the monoclonal antibody industry in terms of improving manufacturing quality, optimization, and not to mention driving down costs. So that was the impetus for ARM undertaking the ACell project. More recently, last year, ARM published AGene, which is also the sister project to ACell, on the best practices on manufacturing of an AAV vector. To make ACell an effective resource and reflective of the ongoing innovation in the sector, we chose to focus on immune cell therapy as a case study. And in particular, we focused this document on autologous CAR-T therapy, given the recent approvals and the number of products that are available to patients in the market. We are very fortunate at ARM to have a large number of subject matter experts from our member companies. There are many contributors for this project from more than 30 ARM member companies. I would also like to thank Nimble, who generously provided funding for ACell, which allowed us to employ technical writers, without which we would not be able to complete the project. The document is intended to benefit the entire field, so it is available as open access in our website. This is what the contents of the document looks like. As you can see, it covers quite a big scope. Each chapter, to an extent, stands on its own, although there is a common QBD theme running throughout. Today's talk will cover the manufacturing principles and examples laid out in Chapter 8. The webinar today is the second out of our 7-part webinar series for the ACell educational rollout. There are 6 one-hour webinars scheduled roughly monthly until April next year, covering various chapters of the document, and it will culminate in a workshop in May or June next year. The schedules for the webinar will be posted in this link, and the registration link as well will be updated as we schedule the upcoming webinars. Our hope is that ACell can serve as an educational tool to inform on the basics of cell therapy, but it will also inform on the methods used to design and quality. As the industry is growing and many people are coming in rapidly into this field, there are a lot of experienced individuals from pharma who are entering the field who may lack the knowledge of the particular unique challenges of cell-based therapy manufacturing. We hope that this document will be used by product developers, educators at universities, and we also have the aspirational goal that this will be used to help educate the regulators. So before we move on, just one housekeeping item. You can use the Q&A function to ask questions, and we'll try to get to as many questions as we can. If we didn't get to your question, we will compile them and post the response in the webinar website. And with that, I would like to introduce our expert speakers for today's webinar. Dr. Fabio Fachin is the head of cell therapy process development, automation, and digital technologies at Takeda. Prior to Takeda, Fabio was at Novartis, where he contributed to the development of Kymriah, and at Torque Therapeutics, where he was VP of Development and Manufacturing. He holds a postdoctorate from Harvard University School of Medicine and a Ph.D. in aerospace engineering from MIT. Prior to transitioning to biotech, Fabio's early career was in the aerospace sector, where he developed microtechnologies for aircraft and satellite guidance. Our second speaker is Bruce Thompson, who is currently the Vice President and Technical Lead for the Cell Therapy franchise at Resilience. He has over 15 years of CMC strategy, product development, and cell therapy manufacturing experience. Most recently, he was Vice President in Process Science at Lyle Immunopharma, where he was responsible for process and analytical development, as well as lentiviral vector sciences and tech transfer of processes. Prior to Lyle, Bruce served as the Senior Director of the Therapeutic Products Program at Fred Hutchinson Cancer Research Center, where he led GMP manufacturing of cell and gene therapy products. He was responsible for supporting over 15 active clinical programs and contributed to the filing of 6 INDs for various cell therapy programs. Bruce received his Bachelor of Arts in Biology, Master's in Biochemistry from The Ohio State University, and Ph.D. in Microbiology and Immunology from the University of Louisville. He completed a postdoctoral fellowship at Washington University in St. Louis in the Department of Pathology and Immunology under Dr. David Fremont. Fabio and Bruce, thank you again for taking the time out of your busy day to be here today. And with that, I will turn it over to you, Fabio, to start your presentation.
Great. Thank you, Josephine. Perfect. To share right now. Great. Well, thank you again, Josephine, and welcome everybody. I think both Bruce and I are quite excited to be here today, telling you a little bit about ACell and specifically Chapter 8. So what we will do today is aim at covering a little bit of the key considerations for development and manufacturing as it relates to cell therapy. But most importantly, in the second part of the presentation, Bruce will take us through and apply those considerations to a mock example that hopefully you will then be able to apply to your own experience and application. So to actually get us started and introduce best today's seminar, I would just actually to start introducing the mock example so that we can all prepare ourselves, if you want to put it in the same state of mind. So for just a second, let's imagine that we are a new biotechnology company that has in-licensed a new construct for an autologous application. The task at hand today, as we talk about the seminar, is that of designing the process to manufacture this product in an efficient and cost-effective way. And you know that it's a product that it's a CAR-based therapy for B cells. You have received your lentiviral sequence for production, but you have enough material to get started. And it's for hematological cancer, so you have some previous information regarding dose and what scale you should be. So Bruce will go in depth into this story later, but even right now for the introduction at a high level, there are some key words and key sentences here that I think we can start using to piggyback onto what Josephine was saying about the seminar series and explaining how this story becomes her holistic view as it relates to the seminars. In the last session, Dr. Cicalese covered very well the topics of QTBP, risk assessment, and CQA identification that are associated with new therapies. And we will assume this underlying, if you want to call it work, as the basis of the discussion today. We will not have the time to discuss those again, but we will take them as, let's say, granted and at the base of all that we discuss. Similarly, there are many other aspects of the story that I just shared, such as the lentiviral manufacturing, materials, analytics, and of course the control strategy that we will not talk about today, but I really do hope that you will be able to sign up for those seminars as well and see the whole picture coming together. The topic of today indeed is that of thinking about how we can build the best process to manufacture this drug that we just described. And here on the bottom right side, you can see the table of contents for Chapter 8. As you can see, it covers quite some material, from starting material considerations to aseptic processing requirements and all the way to some electronic system considerations. We, of course, do not have time to cover all this today. So in this part of the talk, I will give you a little bit of an overview of autologous versus allogenic considerations, and then I'll dive a little bit into some high-level pointers for both drug substance and drug product manufacturing. You'll see that some of the slides that Bruce and I have are a little dense, sometimes wordy, and that's on purpose. We don't want to go through each line today, but we hope that by accessing the recording and the slides, you may be able to use those pointers and apply them even later on to your own experience. So with that, let me jump right away into autologous versus allogenic. I think probably the audience here is quite familiar with this topic. The picture here on the right is actually something that we put in the manuscript. And Simplistically, an autologous therapy is one that is patient-specific. So cells from the individual themselves are collected, engineered, expanded, and then re-injected into the same patient. This is by far still the most common approach in the field for many reasons, but one of them definitely being kind of safety aspect and with low to no likelihood of rejection and graft versus host disease because of the patient-specific nature. The part that we will discuss today in terms of guidelines is how, however, an autologous approach exposes you to intrinsic patient-to-patient variability that needs to be controlled, but also the fact that typically in the autologous space, you start your development using healthy donor materials. However, when you move to clinical manufacturing, of course, you're using diseased patient materials from the patient themselves. And a key challenge is that because those two are quite different, you can't quite fully predict manufacturing outcomes based on your development studies. So you need to plan ahead to be able to react kind of as your trials are ongoing, and we will touch just a little bit on that on the next slide. One last thing that I want to mention about this slide here, even though today we cannot really touch upon it, is that allogenic is, of course, the other side of the coin compared to autologous. It's a patient-independent therapy where typically you take cells from a healthy individual and then you engineer, expand them to then serve a much larger number of patients. This does provide advantages, better control of starting material, potential larger scale economy, but of course there are safety risks associated with this approach, and that often results in more stringent both process analytics, but also especially QC steps that we don't talk about today, but the manuscript does cover. Let me take one more second to discuss this concept of planning ahead. And the reason why that's important, especially for autologous, is because planning for success simplistically means planning for failure. In the next part of the presentation, we will talk a little bit about how we design a potential workflow, such as those that I show here. But the point of this slide is, again, to remember that for autologous, typically development starts from healthy donors, and you develop this process that, however, at some point, hopefully kind of gets put into practice in clinical manufacturing with your diseased patient material. And this is where the field has started to see issues. For example, you could have low yield after a selection step, poor expansion due to potential kind of exhausted cells, and low viability maybe pre or post thaw. And the challenge that you have to take into account is how can you react? How can you solve when kind of accessing kind of the materials, the actual representative materials that you will need to reperform this development, are scarce or impossible to get because they're patient materials. So a concept that is discussed in the manuscript, especially in the expansion section, is that of small-scale models, okay? Where small-scale models are essentially an equivalent process that is, however, using much smaller scale devices and systems, and the development needs to be done still in the context of the QTTP that was discussed before in the previous seminar. The value of developing a small-scale model is that at that point, you will be able to have access to a tool that will allow you to perform these investigations and optimizations on much smaller and accessible patient aliquots. You may be able to get sentinel vials from your collection protocols. You may be able to plan in your manufacturing to collect aliquots, let's say at day 0, or you may have excess materials. The point is that you will be able to actually understand what drove your results based on actual materials that you actually otherwise would not have access to. So plan ahead. This may also serve as assistance in case of deviation and backup in a data-driven approach. And so that's a very key concept that, we discussed in the expansion part of the manuscript, but it applies across the entire operations. So with that, let's jump a little bit into some considerations regarding large scale. And we will guide quickly through both some DS and DP points as we go through the slides. Let's start from the beginning, assuming a cryopreserved product. Let's start from thaw and wash. And the reason why we combine these unit operations is that even though thawing is, of course, kind of responsible for making sure that a sample is controllably and rapidly thawed, it is not sufficient to just stop there. It's important that we remove the cells from the cryoprotectant as fast as we can, because otherwise they may result in toxicity. So kind of as you design this combination of operations, design your thaw strategy, A, to provide enough materials based on what you expect your downstream losses to be in the wash step, which will always unfortunately occur. But most importantly, make sure that the timing from your thaw all the way to the end of your wash, when the cryoprotectant is being removed from the cells, is taken into account. Typically 30 to 60 minutes is kind of what the industry is seeing right now. And the key part to take into account in this regard is that the GMP timelines are very different from those developments. So it always takes longer in GMP. So plan for this ahead of time and pick your systems to provide you the correct throughput to achieve these outcomes. When it comes to the systems available to perform these operations, there's many out there. There's multiple dry thawing devices available in the industry. Many of them use either a fixed thaw time approach, or there are some with active detection of end conditions available as well. In terms of wash volume reduction systems, there's many physics have been now deployed to achieve different instruments for different scales. One key point that I want to stress for the thaw and wash steps here that we are discussing is that throughput should definitely be taken into account, but also make sure that you select a device that will give you a low enough output volume so you can get a concentration that is then compatible, therefore high enough for your downstream steps, which are the selections which typically are very sensitive to concentration. So you avoid the need to do additional steps or additional volume reduction steps. When we move to the selection, the selection is definitely probably one of the most important steps across the entire process. And that's the reason, or that's the way in which we can actually remove the variability, especially for autologous that we discussed, that we observe between patient to patient. So it's very critical as you define these steps for your process, that you define upfront what are your target cell populations and your impurities, the cells that you want to remove, and plan your selection strategy based on that. One key thing that often gets neglected or that results in issues is that you need to plan for the approach where you may need to do one or two or multiple selection steps. Typically, a coarse enrichment, for example, is utilized to, for example, enrich for PBMCs while removing red blood cells and platelets. But then kind of oftentimes, there is the need to kind of go for a finer enrichment, for example, targeting CD3, 4, and 8 or other subsets via a magnetic bead approach. It's important that you design these two steps as one ecosystem because, again, especially the max approach does require specific concentration going in. So make sure you avoid the need to do additional intermediate steps that would result in more cost and longer timelines for your process. Here again, the instruments available are quite a few. In terms of key balances that you need to take into account is purity versus throughput. Those two are typically, one the opposite of another. And then kind of, again, look at the different physics and kind of understand really what type of separation you will need, whether it's coarse or more refined, kind of going from max to also fax approaches, and plan for multiple solutions in tandem. The next step that we want to talk about is activation and engineering. These two are also two steps that we put together for the very reason that the activation step is actually typically required to achieve optimal engineering. The activation function, as the name says, is that of activating and starting the proliferation of the cells, which indeed is needed for the subsequent expansion steps. But in general, you do need to plan for a certain activation time before you can do proper engineering. So kind of as you think about that, select your activation strategy based both on the biology, the cells that you are targeting, but also take into account the process downstreams, both in terms of activation time, but also there is more and more approaches right now in the autologous space that are trying to go to a shorter and shorter manufacturing time frame. So some, for example, bead-based activation schemes make it very hard to remove those beads at the end of harvest if you haven't given enough time for the cells to duplicate. So that may not be something you want to do in that case. So please take this as an overall ecosystem design so that then you can perform your engineering steps also as efficiently. When it comes to engineering, there is many approaches right now. We'll talk a little bit in the next slide, but there's both viral and non-viral approaches. The key call out that we want to make here is that this is one of those key steps where cost is definitely a consideration. You will need to balance for the specifics of your product. How many cells do you want to transduce? For example, versus, kind of the cost of that vector. Ideally, you would like to transduce as few cells as possible. So you minimize your vector cost. However, they may require, additional expansion downstream, which could potentially exhaust your cells further. So that balance between how much vector, how many cells you engineer versus kind of how much you will expand downstream is a balance that is specific to each product. And you will need to take that into consideration for the specifics of your program. When it comes to activation and engineering, there is, again, a variety of approaches out there. When it comes to activation, monoclonal antibodies bound to a substrate are typically the solution. But there's also kind of other reagents such as interleukins that are used kind of in conjunction with those to support expansion. When it comes to the transduction, one of the most common approaches is still viral-based transduction, whereas lentiviral, like in today's example, or retroviral. But there's a plethora right now of non-viral approaches that are coming to the foreground, such as CRISPR-Cas9, TALs, and others, including the instrumentation such as electroporation associated with them. So the manuscript kind of goes quite in length on the engineering side, and we really hope that kind of you can benefit from the material that was included in there. The next step is expansion. And here, of course, the goal is that of achieving sufficient expansion level to meet your target requirements. It seems easy to say, but as I mentioned before, that needs to be taken into the context of the biology of the cells. You do not want to overexpand. So it's very important that you both think about, kind of the steps before, but also think about the steps downstreams and kind of make sure that you plan for expansion levels and yield that will account for the losses that you will observe during your harvest and fill finish steps. The expansion is discussed in depth in the document, including kind of all the instrumentation. And one thing that we want to call out regarding your selection for your systems is scale and clinical stage. Typically, a shorter process or a smaller scale lends itself more easily to manual and maybe 2D culture systems. However, as your culture becomes longer and your scale becomes larger, you do want to start incorporating automation for Ph.D feeding controls. And so you will need to kind of make that assessment based on your program. The part where clinical stage becomes very important is that change is hard as you move forward in your trial. So plan ahead. The timeline gain that you may gain in your phase 1 by going with a manual system may be a much bigger hit, kind of when you cannot demonstrate comparability later on in your phase 2 and beyond. So kind of keep that into account as you're making your choice of reactors for your specific program. Here again, we have a couple of callouts that hopefully will be helpful for all of you in terms of kind of just technologies that I want to call out. When it comes to 2D manual systems, we have a variety of flasks, G-Rex, and bags available in the industry. One of the key limitations of the systems is that integration of PATs is not that easy. So kind of as you think about getting more data and more control of your process, 3D automated systems such as stir tanks, rocking reactors, and others do allow for that compatibility, and that integration is becoming more and more. So as the field goes to a potential quality by control approach, please also keep that in mind because that will be a key component, kind of that ability to use your data at collection to better control your processes. The final step, kind of before you kind of get to your DP, is of course the harvest. And the harvest is a combination of steps that kind of can vary very much depending on all the approach that you have utilized upstream of this. Simplistically, the harvest main goal is that of removing impurities, additives, and other non-target media components, such as, for example, beads, while making sure that you don't lose the cells that you have expanded. So those two are, you may say simple tasks, but in reality, kind of takes quite some considerations to make sure that you achieve this without compromising your cell quality. The two, I would say, functions that, independent of the use of one activation scheme or other, you will always need to perform is a media exchange, where you kind of want to remove the cells from their culture media to a basal preformulation media. But also you want to make sure that you reduce the sample volume enough to be compatible with the downstream requirement. This is really important. You want to avoid to have to repeat a wash and a volume reduction. So please make sure that you design your harvest steps, keeping in mind the concentration requirements of your downstream processes. Here again is one of those situations where you need to plan for multiple steps in conjunction. Depending on your activation strategy, you may need to perform a debeating step that is then being followed up by a wash volume reduction component. And so plan for those. And especially as it comes to the wash volume reduction system, I just want to have a last call out in terms of technologies. Here is where probably you want to really make sure that you select the right technology that will help you both at the beginning, post-thaw, but especially try to pick a device that is also going to be able to sustain your harvest. Here is where your cells are being expanded. Typically, you have a much larger volume, so you need to plan for a throughput and a maximum capacity that will be able to accommodate the entire span of your process. So you need to avoid validation of two different devices in your GMP environment. The minimum output volume, as I called out, is quite critical. You want to make sure that your system gives you a volume low enough so that your cell concentration can be high, avoiding the need for additional spin downs and potentially even, the risk of having to trigger a manual, backup if you cannot achieve the target concentration. So that's quite a key thing that you should keep into account as you think about this step. Finally, because before I pass it to Bruce, I'm going to have one last call out on the DP. The DP would definitely require by itself a dedicated workshop, but we're going to try to quickly kind of touch base on this because it's quite similar to some extent to the discussion that we had about the thawing wash, just on the other side of the coin again. Kind of here, the key function of the steps combined is, of course, the control addition of excipients to stabilize the final cell drug product. Again, here we assume that we're going to go for a cryoformulated approach to then aliquot that pool across one or multiple closed containers, verifying the absence of impurities and labeling, and then in the case of cryopreservation to freeze that sample. The key part that we want to stress, and that's kind of why, again, we put them here together, similar to what we've done with the thawing wash, is that this entire ecosystem needs to be planned as a whole unit. During your formulation step, you always want to keep in mind material compatibility all the way from the beginning to the end, but also you want to make sure that once you have added your cryopreservation, you do take into account the timeline that it will take to get to your final frozen state. There's a limited window, so kind of all your filling requirements, finish, optical inspection, all that time will count towards the maximum that your cells will be able to sustain. So kind of as you define your choice of container, bag versus vials, as you decide your choice of optical inspection for the different containers, as you decide your techniques for labeling, please keep in mind that that time is going to be much longer in GMP, and that may result in very detrimental effects to your cells. And a lot of the issues about post-thaw viabilities that we have mentioned at the beginning are indeed related to that. So do not discount the impact of time in your development scheme and as you qualify and validate your process in GMP. I'm going to quickly skim through this slide because it's more for a post-read for you. There's a lot of material. There's a lot of things that, again, should influence your formulation, full finish, and cryopreservation development. I'm going to just spend maybe a quick second just mentioning that there are more and more automated systems that allow you to fill and finish your products. There is more automation coming up on the vial size. There's some automation already present and also coming up on the bag size. Those two, in terms of throughput, are quite different, though. So based on your scale, you may be more inclined towards bags. If you have a very large scale, you may be more inclined towards vials. And those two have very different instrumentation and timelines associated with them. So please make sure that kind of you take into account the clinical needs into your choice. And hopefully the manuscript will be able to guide you through some of those considerations quite well. So with that, hopefully those were some key pointers in terms of kind of how we try to set up the manuscript. I'll pass it right now on to Bruce to actually discuss kind of how some of these concepts can actually be applied to a specific case study. And so Bruce, pass it on to you.
Excellent. Thanks, Fabio. I appreciate that thorough review of the chapter and the manufacturing unit operations required to make an autologous T-cell product. So what I want to do is, remind everyone that at the end of the manufacturing chapter, there is a section on electronic systems thinking about your manufacturing execution system, your quality management system, your data management strategies. And there's a lot of good information in there about how you can approach the digitalization of your data. So it would refer you to the chapter there. What I want to do now is walk us through a fictitious scenario in developing an autologous lentiviral transduced CAR-T asset for hematological indication. So one question I would ask here is to consider the following statement. You work for an emerging biotechnology company that has in-licensed a new T-cell therapy construct. Your role as the lead of this group is to map out the best process to manufacture this new product in the most efficient, cost-effective way. From the preclinical perspective, the research team has provided a little bit of information, but not enough really to understand what you really need to do to build this. It's a chimeric antigen receptor targeting a novel receptor on B cells, so-called the other B cell receptor, TOBCR. I haven't trademarked that, so it's still available. The research team can provide the sequence, so you have an understanding of the lentiviral production requirements and a tiny bit of research grade vector to get you started. The indication is a hematological cancer, so you can assume some commonalities with dosing ranges and some of the safety elements for liquid tumors. So with that in mind, I want to walk through how we would approach the development aspects. And one of the things that we have the luxury of with the ACell publication is the other chapters, one of which talks about the control strategy and the target product profile. So starting with the end in mind, as Fabio mentioned, is important. What we would want to do here is really understand the intended target product profile and add the quality elements to it. So you understand the therapeutic indication. Your shelf life for an autologous therapy is, half a year to a couple of years, depending on whether you're thinking about redosing, which isn't common but is done. Your storage conditions, whether you want a shelf life stable 2 to 8 and/or cryopreserved product. Container closure, as Fabio talked about, is very important. Whether it's bag or vial based, whether you're thinking about a phase 1 dose escalation, which might require flexibility, or looking forward to pivotal or commercial, in which you understand better volumes and presentations. Your dose regimen, is this a daily administration? Most likely not. Is it a single infusion? Often the case. So that will guide your drug product presentation, your yields for the manufacturing process, et cetera. Other things like the route of administration, how simple is it to get it out of the bag or the vial? What are your general chemistry attributes, pH, osmo, et cetera? How are you going to approach appearance? If it's in an opaque vial and it's an opaque solution, you're looking for visible particulates. You're not going to see any subvisibles, et cetera, because your cells, those are going to be very difficult to detect. Safety aspects, identity aspects, your common testing aspects as well, all need to be considered as you think about the manufacture of the process. So these are some early considerations that we want to think about in terms of the manufacturing process. Fabio walked through these major unit operations earlier, and I'm just going to highlight a couple of things to think about. So for a typical CAR-T, we know we need isolation to get the right product subset of T cells in this case, an activation step to open the chromatin and allow the gene engineering to take place and proliferate the cells to get a sufficient dose to put back into the patient. Again, the engineering through transduction in this case, expansion to get to the dose requirements, harvest and formulation, so ensuring the appropriate wash steps to remove any residuals, cryopreservation and the ability to create the final product in the container closure suitable for the product, and then the reinfusion strategy. So some decision points that need to be made along the way there that I think are informed by the overview. Is this a specialized starting population that's required for this product? Is it a subset of T cells? Is it a CD4, a CD8? Are there memory subsets that are required, because that will impact your selection step? Does the dose require significant activation and expansion? If so, do you understand the yield requirements for in-process tests, for QC retains and release testing, and to meet sufficient dose elements? How will you introduce the transgene? In this case, we said by lentiviral transduction, so that's straightforward, but there are lots of other non-viral approaches that could be taken. Again, your expansion and yield requirements will be dictated by your dose. If it's an unknown scenario, then you think you have to have one of two approaches. Either you have a process that's scalable during your phase 1 expansion, which would require IND amendments potentially, or you build to the largest yield requirements, and then you have lots of material left over for your low dose cohorts. Is there an intervention required? So purification or other treatment at the end of the process. Again, you may not select on the front end, you may select on the back end to either remove contaminants or increase the purity of your desired population. What are your formulation and cryopreservation needs? Again, is this a standard sort of DMSO 10% cryostore approach that is tried and true, or are you looking for something that has a different impact? And finally, are you thinking about redosing? And this is more of a yield requirement than anything else. Your biology may preclude this if you have, a murine scFv that might cause antibodies and clearance. But if you have the opportunity, you need to think through any yield requirements for redosing. So other considerations for optimization of the product, thinking through the characterization of these starting materials, especially if you need a well-defined starting material. In your expansion step, thinking through serum-free media, do you need to, can you remove human serum? Do you need particular elements to support the biology of the product? And that might impact your choice of medias. The expansion vessels, again, you have a number of different options that could be deployed to meet your dose and yield requirements. Harvest and formulation, you can do the old-fashioned centrifugation, media removal, wash, but that's very difficult to scale, and it is an aseptic challenge. So removing those open steps and looking for closed harvest, wash, and formulation options are always good to consider early in development. Cryopreservation, again, considerations around your cell concentration, bag versus vial, your thaw stability. We often see cells that are thawed at time of use, greater than 80% viable. But after that, I think is an open question sometimes because cells are pre-apoptotic and may or may not survive in the same fashion, depending on how they're treated. Finally, stability study requirements, not only in use, but also product shelf life. All of these need to be thought through, and you need to consider which material you might deploy for these studies. Is it an engineering run? Is it a healthy donor? How representative of the patient material will that be? So thinking through that in early development is important. What else to consider? So again, I think there's one thing in cell therapy that is unique and sets it apart. I think, well, there's lots of things, but one thing that's important from a CMC perspective is the impact of timeline and raw materials. So engineering of a T cell product requires, in this case, a lentiviral vector, which requires plasmid containing the gene of interest to be created in the lenti. So you have a linear manufacturing process of multiple raw materials that take time. I think a lot of companies that are new to the space and a lot of individuals who are transitioning in don't really appreciate that once your construct is locked, you have quarters and quarters of GMP manufacturing timelines to initiate and manage because of the raw materials. For autologous cell therapies, chain of identity and chain of custody is critical. Obviously, patient material coming from a patient needs to be engineered, controlled, and given back to that exact same patient, given the safety concerns for allo mismatch safety elements. I mentioned the electronic manufacturing execution system, LIMS system, QMS, et cetera. This is an early consideration, especially when you think about increasing your throughput, scaling out, decreasing your turnaround time. The use of electronic systems will be very helpful in your managing of all of the data associated with each individual batch, associated with each individual patient. Understanding your sampling requirements, so in-process release, retains, characterization, all of these things become important because they are a hit in yield to your final product, and you need to ensure that you have enough material to dose the patient, but also enough material to do the appropriate testing. And I would say that, Fabio mentioned shorter and shorter manufacturing times. That may change your sampling requirements, right? You have to be thoughtful about why you're testing the product, when you're testing the product, and what you expect to see, and how you're going to manage changes. And all of those will impact your sample plans. Finally, always consider collecting more data than you need, because this helps you define your design space and ultimately will help you build a better control strategy. And at the end of the day, your phase 1 process is likely to look different than your commercial process. The more you understand your design space, the better you can approach comparability and late stage changes with data in hand. So always think through with the end in mind, which is a product that may look different, and have the ability to look back at data and say, this is a data-driven decision based on the data we generated in phase 1, phase 2, and pivotal. Okay, so what does this look like in practice? And this is my final slide, but it's meant to aggregate everything that we've been talking about today. So as you think about each of these unit operations, and it is an iterative process. So Fabio went through the overview. I went through a lot of things that are reminders of the overview, and now we're going to put it in practice. So for cell isolation following the leukapheresis, considerations like cell sorting, magnetic separation, or even bulk simple isolation of T cells from monocytes and other leukocytes. Understanding the biology requirements of the product can guide you into the cell isolation choice that's required for the product. Activation, we're talking about a CAR T, so your options here are relatively limited. You're going to engage the CD3 and CD28 co-stimulatory pathways. There are other things like CD2. There's some other co-stimulation approaches that are becoming better known, but you need to make an early decision on how you want to activate those cells. There are reagents that are made today that activate CD3 and CD28 to different extents. Do you need massive proliferation? Do you need a small amount of proliferation? Your choices in that activation space can impact that. Transduction, again, for our purposes, lentiviral transduction, but there is other options for non-viral electroporation, other ways to insert genetic material into the cell, provide that editing gene engineering capability. Expansion, again, scale and complexity here are important. If you need a couple of days and a couple of rounds of expansion, something simple like a G-Rex could be fine. If you're looking at an engineered TCR product, which we're not, but if we were, we might need billions and billions of cells. So we need a larger wave or Sartorius bioreactor to get us to those volume and yield requirements that would then maybe require perfusion or additional media changes. So having the right equipment to accomplish the scale and biology is important. Harvest and formulation, again, thinking through aseptic processing, end-to-end, closed cell washing, formulation, harvest elements are important because you reduce your risk of contamination. You also simplify the downstream activities through these closed unit operations. Finally, cryopreservation, there aren't a lot of great options, right? So, if you think about cryo, you think about a controlled rate freezer, you think about a small number of formulations, and you think about storage and liquid nitrogen. There are some new electronic options that replace some of the liquid nitrogen-based CRFs, and those are gaining in popularity. So being thoughtful about how you want your final product to look. Do you need lots of throughput? Do you need a small number? Do you have the available liquid nitrogen capabilities in your facility? Those are all decisions that you need to think through as you design that process. So I know I went quickly. I wanted to leave time for questions and answers, but I wanted to also walk you through how you may utilize all of this information in a practical way to set up early engineered T-cell development aspects. And with that, I'll hand it back to Josephine and we can start the Q&A.
Great. Thank you so much, Bruce and Fabio, for a fantastic, very comprehensive presentation. So now we have just a little over 15 minutes for audience Q&A. And again, as a reminder, please use the Q&A function and not the chat. So maybe Fabio and Bruce, as we allow the audience to type in their questions, maybe I'll open with one. So I think you both mentioned the importance of start with the end in mind. But you also hear about the importance of having the right size development program for your immediate needs. So as you think about scale up, you don't want to start your early stages with a 3D automated expansion platform right away. So could you share your strategies on the timing? When do you want to make that switch, and when should you start your comparability studies? Maybe I'll start with Bruce for that.
Yeah, thanks, Josephine, for starting with not a softball for sure, but we'll get the discussion going. So there are a lot of variables here for process changes, right? And the comment that I made around generating sufficient analytical data to understand your control space is important. to me, every time I approach these things, it's a mental risk assessment around, is this a change I need to make prior to phase one? Or is this a change that I'm comfortable making in a pivotal setting? And so if you think about a scaling impact where you're changing the duration of a manufacturing process, that might have an impact on your T cell fitness. We know about exhaustion. We understand more about the biology today than we did a number of years ago. So we might be thoughtful about assessing that earlier in development versus making a change prior to pivotal. Thinking about technologies, there's always a challenge because programs that are in development often have associated clinical data, and that clinical data in many of these cell therapies is quite impressive. The efficacy of a lot of these products is high, and therefore the concern about making changes that could impact the efficacy is also high. And so again, the better we understand the product, the better tools we have to measure change, manufacturing process changes, and the impact to the product and its efficacy and safety, the better we feel about making those changes. So the answer is kind of complicated, right? It is, if you don't understand your product, you have an aversion to making these process changes. If you do understand your product, you have an easier path because you know how to generate the data to support those process changes.
Great, thank you, Bruce. Fabio, anything to add?
No, I would say I quite well agree with what Bruce was saying. I think it always comes down to data. I would say for the specific of an expansion, you're going to mention the bioreactor, that is definitely one of those that gets categorized under the bucket of major changes, kind of similar to transduction and others. So those are definitely aspects of your manufacturing that are harder to change later on in your trial. So I think where I would maybe add something to kind of what Bruce said is there are right now technologies that are becoming a little bit of an in-between, without kind of going into the details, but for example, stir tanks, there are options that allow you to have, just say a smaller scale, then you can kind of scale up as your let's say program moves forward. So I would definitely encourage, the audience here to consider those. But in general, it's one of those where if there's a chance, I would try to plan for success earlier for these major changes. So kind of, if possible, try to incorporate as much in phase one and early trial stages, because again, you may incur the risk that even when you have the data, you may still not be able to show comparability, against your original system, and therefore you may not be able to move away from it, despite kind of all the data you have at hand. So.
Okay, great. Thank you, Fabio. Okay, so we're starting to get some audience questions here. I'll direct this one to you, Fabio, about fill and finish. So do you see more and more the need to perform fill and finish in 2 to 8 degrees Celsius environment to reduce cell viability issues?
Yeah, it's a great question. I think, of course, is going to be one that it's program dependent. I mean, some therapies do, kind of, let's say, cool down the cells, maybe, pre-cool reagents, etc. Some other approaches I've just seen that in room temperature operations. I would say there's definitely some, data that's showing that cooling down the cells does help, if you want to call it, the stability, or at least in process stability. So I think there's benefit. The key challenge you're calling out with your question there is, the way I'm reading is the environment, right? What are you actually trying to control? Are you trying to control, if you want to call it, let's say that you do this as an isolator, the temperature within the isolator, or are you actually trying to control, the cell volume and fluid per se? I think that if it's the former, then there's definitely, kind of tools that are kind of coming up to do that. Being able to actually actively control the temperature of the fluid, I think it's a little more challenging. I do think that there's benefit there, but my opinion would be, unless you have data that shows that you really need to actively control your sample temperature, I would first kind of try to default to the easier route. But then, kind of, if the data really support it, then kind of embrace that. And again, the field is going in the direction with some tools that allow you to do that. But that will be kind of my my first answer there. Let's let the data drive that decision. And again, if you're able to design your processes to be short enough, then the advantage of having to cool down actively your substrate it's a little less relevant. So I think it's something you will need to kind of look at on a case by case.
Great, thanks, Fabio. Yeah. I guess while we're on the fill and finish topic, can I just follow up? Do you have any strategies on? Could you share a strategy about qualifying bags or kind of other plastic consumables that you use in the manufacturing? So are there testing responsibilities that fall to the drug product manufacturer that isn't part of your release or stability programs? So do you have to perform kind of your own leachable extractables with your product? Could you share maybe your experience with that? Either Fabio or Bruce, you want to take that? Yeah.
You want to take this, Bruce, from a...
Sure. I mean, I think there's always a remit for the sponsor to understand the elements of anything that comes into contact with your therapeutic product. And so I think the extent to which a lot of the residual impurity assessments are done for early phase is relatively low. I think we understand the risk-benefit profile, but as development progresses, I think there should be an initiative to better understand your product contact elements. As an industry, we've begun to progress. We have a number of new technologies that are being brought to the market or being introduced into the patient settings. So I think there will be a maturation of our understanding of those product contact elements, and there'll be better assessments for materials of construction, for any residuals that might be present in the packaging, plastics, consumables, et cetera. I think we'll continue to get better at that. But again, as a sponsor, I think you need to do the safety assessment and look at your product and ensure that you're not introducing something that would cause harm in clinical development, and then understand how to mature that as your product development progresses.
I would agree. And then kind of just to add, I think at different stages you were asking, kind of, does the sponsor have the same responsibilities? I think across the different stages of your trial, okay, you will have different requirements. Like at the beginning, maybe in phase 1, for some things you may just perform a paper risk assessment that may be sufficient, but kind of as you move forward, kind of you'll be required to do more and more testing. And I would say also kind of if you are introducing, if you have a custom technology or a custom kit or a custom disposable, in that case, your requirements for testing extractables and leachables is of course greater than if you have an off-the-shelf technology that already kind of has documentation associated with it. So I think that's also a difference.
Okay, great. Thank you both. We have a question about transfection technologies. So are non-viral transfection technologies likely to become accepted or prevalent in the industry, especially with some of the challenges around electroporation? So who would like to take that first?
I can take a shot and then hand it over to Fabio. I mean, I think the answer is yes, right? Because there are cost elements, there are availability elements, there are supply chain elements that would suggest that if every product in development requires plasmid production, vector production, and all those accompanying release tests, et cetera, the cost for these therapies will continue to be high. So looking for alternatives to introduce, gene engineering will always be a driver of innovation. There are safety questions, right, around editing of genes, and the autologous space. I think the risk benefit maybe is different than in the allo space, and that's something we're learning through the biology as well. So again, I think as our tools to assess analytically, the genomic changes continue to progress, we'll see more and more gene editing elements introduced into these therapies. So, electroporation, we're seeing new instruments from vendors, on either in production today or on the near-term horizon. And the more efficient we are at introducing genetic materials to cells, the more uptake we'll get in that gene editing space.
I would agree. I think there's definitely, as every industry, at the beginning, inefficiencies are there, and this is one of those kind of where the technologies are still in the early phases. I mean, I would say that applies probably in general cell therapy, but definitely kind of this non-viral approaches are one of the newest fields, put it this way. There are already other alternatives to electroporation that are coming up, mechanoporation, there's chemical transfection, etc. So I totally agree with what Bruce is saying. I think the field will keep evolving. There is clearly an economy of scale that I think it's playing to the advantage of this further advancements in technology that hopefully we will see. But the technology, unfortunately, kind of is to some extent decoupled from the analytical point that Bruce is mentioning. So I think as long as the data in terms of the safety of these therapies will pan out, I do believe that there's going to be technologies that will allow us to then deploy them efficiently across patients. So I think both are going to go on in parallel, and at this point, I don't see any reason why the allogenic field, shouldn't move forward and the technology with it.
Okay, great. Thank you both. Yeah. So the next question. So as you think about lot size and scale, could you share best practices on your assumptions related to typical losses between processes or unit operations?
Ooh, okay. That's one of those where we should do some math. But I would say, in general, maybe I can, take a first step here, kind of, Bruce. And I would say, if I go quickly through the process, I would say that if you optimize your freestyle cycles, probably about 10% loss is a good expectation for an average freestyle cycles. As you think about washing operations, kind of where you're really trying to just retain all your cells, but just perform, let's just say, a media exchange. There I would say your range is probably going to be somewhere around 80 to 80%. Kind of depending on your materials. There's definitely a chance to go higher, but it will also depend on the balance with throughput. But I would say assuming 80 to 100 would maybe 85-90% mean, I think that would be possible. When it comes to the selection steps, that's a little harder, right? Because it really depends on kind of what you're trying to remove versus target at the same time. If you're trying to go for a positive selection approach, then I think you probably can expect somewhere around 70 to 80% recovery. But if you have very high purity requirements, I think that it's a little unreasonable to expect beyond that. All the other operations I feel that are probably kind of along the same kind of range, but kind of if you then think about kind of how this maps end to end, and that's kind of why throughout the talk we mentioned try to avoid having to do intermediate steps, because that 70-80% recovery or the 20% loss really then kind of exponentially kind of impacts you as you start to kind of piece together all the different operations. So, but I would say that those are probably good guidelines. I don't know, Bruce, if there's anything else in your experience that you would add.
Yeah, no, I think Fabio, you captured probably the overall general recoveries. I think the one thing I would say is, as we understand more about the biology of T cells, we do need to pay attention to whether those recoveries are biased, right? So thinking about activated T cells being a little bit larger, are you selecting or sort of inadvertently deselecting different populations based on some of these techniques and unit operation approaches? So just paying attention to the impact of not only the recovery, but the biology associated with those cells that are recovered is important.
Great. All right. Thank you both. All right. We have 1 minute left, so we have time for one more question. Could you briefly touch upon cryopreservation of the leukopak itself, so upstream of the thaw where you started, Fabio? Do you have the same considerations there as the cryopreservation of the final drug product?
Well, that's a very good question. I would say it's a challenging one too. I think my first guess here is my first answer would be really make sure that you identify the right clinical partners and work with them. And that's kind of the reason why I'm saying that is typically kind of when you do your development, for example, in the US HemaCare is a very, kind of standard. You get a supplier, you get a frozen leukopak from them, but you don't really know the, if you want, background of that freezing protocol, to some extent, or the processing. However, you do have a chance if you want to develop your own. So if you get a fresh leukopak, you can invest internally into then kind of developing the cryoformulation strategy that then you can try to deploy to your clinical sites as you onboard in your own trials. Like one of the challenges going from, let's say, a HemaCare to the different clinical sites that you may have in the trial is that the clinical sites may use very different protocols that are standard in their systems, and that introduces yet another point of variability within your trial. So if you have the ability to standardize your freezing protocol for your specific application and deploy it as a kind of single protocol across your trial, that's definitely something you should do. I think there's not enough time here to kind of discuss the specific of the optimization of a leukopak versus, a bag for DP or a vial. But in terms of kind of key consideration that I would like to take out here is make sure to minimize yet another variability point in your starting material, because otherwise it will just make so much more difficult to kind of get good data and control over your process.
Yeah, I appreciate we're up against time, but I will add that there's some really good logistical elements of freezing either the leukopak or the selected material, right? It allows you to level load your manufacturing facility, allows flexibility in the unit operation starts and stops. And just to harken back to the biology, right? So understanding the heterogeneity of a leukopak versus the heterogeneity of a final drug product, it's quite different. So your thaw and downstream unit operations from a frozen leukopak versus a fresh leukopak could look different. You'll have additional cell lysis, you'll have viscosity issues. You'll just have to pay attention to the additional care required to process frozen leukopaks versus fresh leukopaks.
Okay. Great. So we're at time here. So I want to thank you both Fabio and Bruce again for taking the time out of your busy day for this fantastic presentation, and thank you the audience for sticking around and your engagement in the Q&A. Great questions today. So if we definitely didn't get to all of the questions here. So what we'll do is we'll compile them and we'll post a response on the webinar webpage. And with that, we'll wrap up. So have a great rest of your day, everyone. Thank you again. We'll see you when you're in the upcoming webinar in January.
That's great. Thank you, everyone.
Have a good rest of the day.